It's Biology 160, Introduction to Anatomy and Physiology, talking about the urinary system. In this part of the lecture, we're going to look at the functional parts of the urinary system, which are these little vessels, these little things called nephrons. And this is where everything happens inside the kidney, are these nephrons. So we're going to look at the anatomy and physiology of these nephrons and how they work in carrying out the functions of the kidneys. This is where the urine is formed, and there's a couple of main structures of the nephron. There's what's called the glomerulus, and we'll see this here in just a minute, and then also the renal tubule. And the nephron, basically this is where the blood is being filtered. It's a little filtration device that takes the blood in, filters it, keeps the stuff it doesn't want, and excretes that out into the ureters, into the urinary bladder, and then can put the stuff that we do want to keep back into the blood. That is the function of the nephrons. The kidney is stuffed full of these nephrons. There can be as many as 1 million nephrons in one kidney, so it's pretty amazing. So these are very small structures, microscopic. When you cut open a kidney, you can't see the nephrons. You'd have to get under a microscope to be able to see them. But this is where the nephrons are located. So to get our bearings here, here's our kidney, and we're taking a little cross section out of this kidney, and we have part of the cortex and we have part of the medulla, and that's what we're looking at here. So the outer section here is going to be the cortex, so I guess it shows it here. Here's the cortex, and then here's the renal medulla. Notice how the blood vessels run right along this section between the medulla and the cortex. Some of the blood vessels, though, then run up into the cortex, and then we have our nephron. Here's our nephron right here. This specific nephron is called a cortical nephron because it is contained completely within the renal cortex. So notice it doesn't leave this cortex region. This is how most nephrons are. They're found just in this cortex region. Notice how there's all this tubing that comes off of the nephron that's all convoluted and bent in a lot of different ways. That's where the filtration is going to happen. There are other nephrons that will dip down out of the cortex down into the medulla before coming back up and dumping into the collecting duct, which will then go into, which is where the urine starts to collect. This is called a juxtamedullary nephron. So juxta means to have two things, compare two things. So we have so this nephron is moving from the cortex into the medulla and then back into the cortex. Every nephron has some specific parts that I'm going to point at now, and we'll see them again. And this is true of the cortical nephrons or the juxtamedullary nephrons. First of all, there's the glomerulus. This is also called the Bowman's corpuscle, and we'll see this again in a minute. But this is where the filtration initially begins. And then from there, we have the proximal convoluted tubules. And you can see them here. And then we have the loop of Henle. And then we have the distal convoluted tubules. And then these eventually are going to dump into what's called the collecting duct, and then into the renal pelvis, and then down into the ureter to the urinary bladder. So let's look at some of these parts in more detail here. First of all, here's the glomerulus. So the glomerulus is where the blood meets the nephron. So here you can see the afferent. Afferent means going to, arterial coming in, and then you have the efferent arterial coming away from the glomerulus. Here's the glomerulus capsule, and within this you have these you have capillaries. So this is the first set of capillaries. So the afferent arteriole comes in, goes through these glomerular capillaries, and then into the efferent arteriole. And then it's going to go to the next set of capillaries. But this is the first set of capillaries. These capillaries are covered with what are called podocytes. Covered with podocytes. Podocytes means foot, and these have little foot-like extensions that they cover, and you can kind of see how they're looks like little toes sticking off of these cells. They cover the capillaries, but between these toes you have these spaces, and these spaces allow what is in the capillaries to filter out into the glomerulus. So as the blood is coming in, filtrate is coming out into the glomerulus, which is the first part of the renal tubule, and then from there it's going to go down and it's going to be processed. This is the proximal convoluted tube coming off here. After the glomerulus, you're going to go into the rest of the renal tubule, which isn't very long. It's about maybe 3 inches long, but it's very convoluted, but it's still very microscopic. And this is where the rest of the urine production happens. So here you can see another picture showing all the parts. So we already talked about the arteries, or the arterioles coming in. So here's the afferent arteriole, and you're going into glomerulus, and then you have the efferent arteriole. Coming off the glomerulus, see this tube coming off that's all convoluted, it's all wrapped up. That is the proximal convoluted tubule. Notice how it's associated with blood vessels. That's the second capillary bed. So the first capillary bed here in the glomerulus, and then the blood comes back into the efferent, and then it goes and it enters another capillary bed that is all around this convoluted tubule, as well as the loop of Henle, which dips down here, and then part of the distal convoluted tubule before moving into the collecting duct. This first set of capillaries is designed for filtering, or for releasing things into the glomerulus. The second group of capillaries is designed to reabsorb things. So as the blood's coming in, stuff is filtering out into this nephron, into the renal tubule, and then it's moving into the proximal convoluted tubule, and then in the proximal convoluted tubule, stuff is being reabsorbed. Things that our body wants or needs are being reabsorbed back into the capillaries throughout this whole system, till by the time you get to the end, all that's left in the renal tubule is the waste products that we no longer want. So here the two sets of capillaries are listed out. We have the glomerulus capillaries, and we have the peritubular capillary beds that are around the rest of the renal tubule. The glomerulus capillaries are unique in the body in that they're fed and drained by arterioles. That doesn't happen anywhere else. The afferent arterioles are coming in, and they're dumping off the blood. The afferent arterioles are made to release material out into the glomerulus. An interesting thing though is that the afferent arterioles are larger than the efferent arterioles. So the arteries coming in, arterioles coming in, are bigger than the efferent which are leaving. And what this means, what this causes, is a high buildup of pressure in the glomerulus, and causes everything to be pushed out into the glomerulus. Anything smaller than a protein. So proteins, red blood cells, white blood cells, they don't go out into the glomerulus, but everything in the plasma will. By the time it's gone clear through the nephron, 99% of that's going to be pulled back in, but initially almost everything besides the red blood cells and proteins in the blood move out. In this picture here, you can see how the afferent arteriole is much thicker than the efferent arteriole coming back out. So you have a high pressure buildup, pushes everything out into this glomerulus, where it can then be processed. The process of filtering the blood and creating the urine has three main parts. The first is glomerular filtration. We've already talked mostly about this, but this is where the blood is coming out and being filtered out into the renal tubule. And then here's the efferent blood coming back out. The next step is called tubular reabsorption. The way the nephrons work is pretty interesting. Again, it is a cleaning system, but the way they clean is a little bit different than what you might think. So if your mom asks you to clean your room, if you still live with your mom, she might still have you clean your room. Or if you are the mom, you might ask your kids to clean the room. Or if you're not living with your mom, you might ask yourself to clean the room. But anyway, if you have to clean a room, there's a couple ways you can do it. The first way would be to go in there and just remove the things that you don't want and leave everything else in the room. The second way would be and this takes more time, but it may be, in the end, it may end up having a cleaner room than not. The second way you can clean that room is to go into the room, take everything out of the room, not just the stuff you don't want, but the stuff you want as well. Take everything out of the room, and then look at that pile of stuff, and then take the stuff you want and put it back in the room. Okay, that second way of cleaning, this is how the nephron works. So initially, almost everything is being taken out of the blood, except the stuff that's too big, the red blood cells and proteins. And then through the functioning of the nephron, the stuff that you want is put back into the blood, which ends up being about 99% of what you initially took out. You're putting almost everything back in, but in doing that, you're keeping out the stuff you don't want. So first we filter everything out, almost everything, and then we have tubular reabsorption, where we put everything back into the blood. And this is why it's important that we have the second set of capillaries called the peritubular capillaries, because they're going along the nephron and they're pulling everything back in to the blood. So water comes back in, glucose, sugar, amino acids are going to get pulled back in, and needed ions are transported out of the filtrate in tubule cells and then into the capillary blood. This reabsorption happens almost immediately. Most of it happens almost immediately, and then it happens a little bit more throughout the nephron. Water, glucose, amino acids are pulled back in almost as soon as they're pulled out. All the glucose is removed back out of what we call the filtrate, and amino acids as well. So what this means is that you shouldn't end up with any glucose in your urine. If you do have any glucose in your urine, even though it's initially filtered out, it's all going to get pulled back in, so you shouldn't have any glucose in your urine. If you do, that's going to tell you to have some sort of disorder. Amino acids are the same way. So those are things that are almost immediately pulled back into the blood because your body wants them. And then finally we have some tubular secretion. So after we pull everything back into the blood, there are still a few other items that we push back out into the nephron that we want to get rid of, and these are mainly H positive and K positive, which deal with acid base and ion concentrations in the body. Creatinine and drugs, they're actually removed from the blood and specifically secreted back into that nephron. This is a little diagram here that shows how these processes work here. So initially we have the glomerulus right here, and everything is filtered out. So in the filtrate we end up with water, salts, bicarbonate, H positive urea, glucose, amino acids, some drugs. All of that is secreted out. Once again, blood cells and proteins are not filtered out. If you find blood in your urine or proteins in your urine, again, that's going to be a cause for alarm. So that's why when you go into the doctor, they often have you do a urine test, and they test all these things in the urine because they're trying to see if anything that's not supposed to be there is there. So they test for blood, they test for proteins, they test for amino acids, they test for glucose, test for ion concentrations. If if most of those show up, then that's bad. Ion concentration, they can figure out if there's something going on. And so that's why you do a urine test. So initially everything comes out, but then notice everything that's being filtered back in. So NaCl, your salts, your bicarbonate, your water, your glucose and amino acids are all initially pulled back in to the blood. A lot of this happens passively, but most of it is going to happen actively. So we're going to actively be pushing this stuff back into the blood. It's going to take energy in order to do that. And most of this reabsorption happens in the proximal convoluted tubules. That's what this is here. Notice we also have some secretion going on here too. We're pushing out some drugs and some H positive ions. Then we go down into the loop of Henle, where we continue to reabsorb some things, and this is done by concentrations. And then when we get to the distal tubules, we have some further absorption and some secretion, and then we're into the collecting duct, but we're still not done. We're still going to reabsorb and concentrate that urine in this area here. The red arrows mean active transport, the blue mean passive, and then the green is talking about secretion. So notice salts are actively, glucose amino acids are all actively pulled back in. Bicarbonate ion in this case is pulled back in actively, meaning that it takes energy. Some passive transport happening. Water is passively moves through osmosis. It's going to follow the concentration of water. And then some things are secreted, and that means that they're going to be, some are actively secreted into the urine. And so by the time you get to this collecting duct, about 99% of that initial filtrate, what was initially filtered out of the blood, has been reabsorbed back into the blood, and we are just left with the things that we're really trying to get rid of, and it's highly concentrated. Especially water, we don't want to lose too much water, so in the end we're pulling out more water, pulling out some urea to make that the concentrated urine that we're trying to produce. These are the things that are not reabsorbed in general. Urea, which comes from the breakdown of protein, which is toxic. We get rid of urea. There is a limited amount of urea that's always in the blood, but we try to get rid of that. Uric acid comes from nucleic acid breakdown, and then creatinine, which comes from the muscle cells. Okay, to end up this section of the lecture on how the nephrons work, we're going to watch a little video here that will help to animate this, and hopefully this will make sense here. So we're looking again at the kidneys. This is going to be the left kidney. The right kidney is a little bit lower than the left kidney, and we're going to pull out a nephron, which is the microscopic tubule inside the kidney. So here's what our nephron looks like. Notice we have our corpuscle here. We have our proximal convoluted tubule. It's all, if you could follow this here, you'd see it twist around, and then it comes down into this loop of Henle, and then it's going to come back into the distal convoluted tubule, and then eventually go into this collecting duct over here. We call this the renal corpuscle. Here's the renal tubule. Now we're going to zoom in on the glomerulus. So here's our glomerulus, here's our afferent arteriole, here's our efferent arteriole, and then this is going to be the capillaries with the podocytes. Throw on the top, we call this Bowman's capsule, and this is where the filtration happens. So looking at this little animation here, so the blood's coming in, going around through these capillaries. Now we're going to stretch this nephron out so that the convoluted tubule is not all twisted up, so we can see the pathway of the filtrate. So everything's filtering out, then it's going to move down this renal tubule towards the end. So notice all this stuff filtering out of the blood, and we're moving into the proximal convoluted tubule, down into the loop of Henle here, and then the distal convoluted tubule into eventually into the collecting duct. Now we can see the blood supply that's associated with this. The afferent arteriole coming in, the efferent arteriole coming out. Afferent is larger, efferent smaller. High pressure makes a filtrate. Peritubular capillaries associated with the proximal convoluted tubule, the loop of Henle, and some of the distal convoluted tubule, where the reabsorption is going to happen, and then finally the secretion. And there you go. So this is the process by which the kidneys are able to filter that blood, pull everything out, reabsorb what we need, and then secrete and keep in what we don't want, and then that can finally be secreted from the body.
Thank you.